Here we're going to discuss the process of protein synthesis, which is sort of a continuation of the transcription process that we discussed earlier. So we have DNA being transcribed into RNA inside the nucleus. Then this RNA is going to travel to the cytoplasm, where it can be translated into the protein which is coded on the DNA. The organelle that fabricates or synthesizes the protein is called the ribosome. As we mentioned earlier in the class, ribosomes are made of a ribosomal RNA in addition to some proteins to make up its 3D structure. As I just mentioned, they are the protein synthesizing machines. They bind mRNA and individual amino acid tRNA to the ribosome to increase the efficiency of the translation process. They are very efficient, where they direct the elongation of the growing polypeptide chain at a rate of 3 to 5 amino acids per second. We've mentioned earlier in the course that there are two types of cells, prokaryotic cells and eukaryotic cells. There is a fundamental difference between prokaryotic ribosomes and eukaryotic ribosomes that we need to point out. Ribosomes that are present in eukaryotes are larger in size. They have more RNA chains and more proteins in their structures. They have less subunits. We'll discuss subunits in a bit. And they are simpler. As you know, prokaryotes do not have a nucleus, so translation or protein translation takes place directly after the transcription process inside the cell. However, in eukaryotes, the mRNA needs to be transported outside the nucleus to the cytoplasm, where eukaryotic ribosomes translate it into a protein. If you see here, the ribosomes in both prokaryotes and eukaryotes are made up of two major units, which we call the large subunit on top and the smaller subunit at the bottom. These subunits are made from different amino acids and more specifically from RNAs, ribosomal RNA. We mentioned there is an rRNA or ribosomal RNA that makes up most of the structure of a ribosome. There's the 23S rRNA with the 5S rRNA. They make up the larger subunit in the prokaryotic ribosome, where there is the 16S rRNA that makes up the smaller subunit in the prokaryotic ribosome. Whereas in the eukaryotic ribosome, there is there are three separate subunits, that make up the larger subunit on top, and one subunit that makes up the smaller subunit at the bottom. Notice that all of them have the letter S. This S letter, the 23S, 5S, 5.S, is how they are named. So this is the 50S subunit or the 30S subunit. S stands for the unit Svedberg, which is the unit of sedimentation. We've already discussed centrifugation, and we said that we measure it using sedimentation, and its defining unit is the Svedberg. As I mentioned, ribosomal subunits are designated in the sedimentation rate in Svedbergs. The difference between prokaryotic and eukaryotic ribosomes, as we mentioned, the ones in the eukaryotic ribosomes are larger, so they have a longer RNA molecule making up the subunit. There are more proteins associated with the eukaryotic subunits, and the size of the eukaryotic subunit is larger than that of the prokaryotic subunit. This is a 3D structure of eukaryotic ribosome. The purple part here on top is the large ribosomal subunit or the 50S subunit, and the bottom green part is the small ribosomal subunit or the 30S subunit. Notice here that you have RNAs that make up the rRNA, you have proteins that are also ribosomal proteins that make up the ribosome. Also notice the 3 letters here in the middle between the large and small subunit, the E and P and A over here. These 3 letters stand to 3 different locations in the subunit. Each location has a specific function in the protein synthesis process that we will discuss later on. As in transcription, there are 3 parts for translation, for RNA translation into protein: initiation, elongation, and termination. They both have the same name. And if you remember from our mRNA decoding lecture, we said that every 3 nucleotides are going to stand for the code of 1 codon, which stands for a single amino acid. And we also mentioned that there's only 1 start codon, which is the AUG codon, and that has to be present in order for protein translation to start. There are 2 different methionine transfer RNAs, and the AUG here stands for methionine. There is the tRNAi methionine. This is a specific tRNA for the AUG at the start of the polypeptide sequence, and this is only present at the beginning. Notice that there are other possible methionines down the road after the start codon. However, these are coded for by a different tRNA, which is the tRNA methionine. This incorporates the methionine into the growing chain. So the tRNAi methionine only binds to the AUG at the start of the codon sequence. The tRNA methionine matches the AUG that incorporates methionine to the growing chain, so past the start codon. So only tRNAi methionine can bind in the appropriate location in what I just showed you, the P site in the small ribosomal subunit. Initiation starts at the first AUG at the 5' end of the mRNA. Remember, RNA and DNA have 2 ends, a 3' end and a 5' end. The small and large ribosomal subunit are usually loose and unattached to one another if there is no translation. And there is no translation if there is no mRNA. So if there is no mRNA present, the 2 units are separated in the cytoplasm, and they only assemble once mRNA is present. And it has an amino-acylated initiator tRNA, that's the tRNAi methionine, correctly positioned at the start codon, which, as we mentioned earlier, is the AUG. There are initiation factors that are very necessary for the initiation process to start and to be well controlled so that the correct protein can be synthesized. Initiation factors function include stabilizing the translation complex and proofreading the translation process using GTP hydrolysis and GTPases that we described earlier and we will describe again in a little bit. Initiation factors usually bind to the inactive ribosomal subunit in order to prevent them from binding. This is without the presence of mRNA. The first initiation factors we are going to mention is EIF3. This initiation factor binds to the 40S or the small subunit, and the initiation factor EIF6 binds to the 60S large subunit. Notice that the letters IF in initiation factor stand for I initiation F factor. The E part here stands for eukaryotic initiation factor. If the E is not present, that means that we are talking about initiation factors related to a prokaryotic cell and not to a eukaryotic cell. The same goes to the EIF6. So again, the EIF3 is bound to the 40S ribosomal subunit, and the EIF6 is bound to the 60S ribosomal subunit when they are not in an active state, and they are not attached to mRNA and are not translating any proteins. These initiation factors later on participate in associating the ribosomal subunit once the small subunit is charged with an initiator tRNA or a tRNAi methionine. This is the general outline of translation initiation, and we are going to go through it one step at a time. But in general, if you notice, the 60S and the 40S subunits are not attached to one another at the beginning of the translation process. The 60S subunit is bound to EIF6 to prevent it from binding to other subunits. The 40S subunit is bound to EIF3, and these initiation factors prevent them from binding to one another or other subunits. In order to initiate translation, we start with the smaller subunit attaching to the, as we mentioned, the tRNAi methionine. So this attachment of the tRNAi methionine to the small ribosomal subunit is called the pre-initiation complex, and it has the 40S subunit in addition to the already present EIF3, which itself associates the EIF1A, and this is a ternary complex that consists of methionine tRNA and EIF2 bound to GTP. The GTP bound EIF2 binds tRNAi methionine. So the cells are going to regulate protein synthesis by phosphorylating GDP bound EIF2. The 5' cap end of the mRNA, as we remember, there's a 3' end to the mRNA and the 5' end to the mRNA. The 5' end to the mRNA is usually bound to another initiation factor, which is called the EIF4. This complex, the EIF4 plus the mRNA, associate with the pre-initiation complex that we just mentioned through the interaction of the G subunit of the EIF4 that is bound to the mRNA, and that is bound to the EIF3, which is already bound to the smaller subunit on the pre-initiation complex. This would form the initiation complex. As mentioned earlier, translation initiation starts with the smaller ribosomal subunit that is bound to the EIF3. Then comes in EIF1 and EIF2 bound to GTP and tRNAi methionine, which is also bound to methionine. So we have a complex made of the smaller subunit, the EIF3, the EIF1A, the tRNAi methionine, and EIF2 bound to GTP. This is called the pre-initiation complex. Then comes in the targeted mRNA, already bound to the EIF4, that is bound to the 5' end cap of the mRNA. If you remember, the 3' end of the mRNA has a poly-A tail. You can also notice that EIF4 has multiple subunits, each of which has a task, which we will mention in a bit. So once the pre-initiation complex binds to the mRNA, and that binding takes place by binding the initiation factor 4A to EIF3, that would require the positioning of the AUG sequence in the mRNA at the tRNAi methionine. However, once these are bound, ATP is hydrolyzed to ADP, and EIF1A, EIF3, EIF4 complex, and EIF2 GDP, because the GTP is hydrolyzed into GDP plus phosphate group, are all hydrolyzed once this AUG sequence is recognized by the tRNAi methionine that is bound on the smaller RNA subunit. Notice that this takes place after the formation of the initiation complex. So the difference between the pre-initiation complex and the initiation complex is only that the mRNA and the initiation factor 4 are bound to the pre-initiation complex. The EIF4 subunit has a structural role to put together all the other subunits of the EIF4 plus the mRNA. The positioning of the EIF4A RNA, which is a helicase, so it's an enzyme that untangles any helical structure in the mRNA, so that this helicase removes the short regions of the RNA that has secondary structures using energy from the ATP hydrolyzed in the figure before. The initiation complex is going to scan the bound mRNA, and it's going to stop once the tRNAi methionine anticodon recognizes the start codon, which is the AUG present on the mRNA. So this is the initiation complex. This ATP is hydrolyzed for EIF4A to untangle any secondary structure in the mRNA. And then the mRNA is scanned until the AUG or the start codon is found, and the methionine, the anticodon, is bound to the codon on the mRNA. Once that is recognized, the EIF1 is released, EIF3 is released, and the EIF4 complex is also released from the pre-initiation complex, and we only have, and notice that the GTP bound to the EIF2 is also hydrolyzed. This is once the codon identifies the anticodon. The GTP is hydrolyzed once they are recognized, so that it would lock the location of the mRNA at the anticodon, so that we don't have any frame shifts. So all these initiation factors are released. Then the 60S subunit, or the larger subunit that we showed you was bound to the EIF6, binds to the smaller subunit, and once it recognizes the correct location of the tRNA, it also hydrolyzes the GTP on initiation factor 5. As I mentioned earlier, the recognition of the start codon leads to the hydrolysis of the GTP associated with EIF2. This is to prevent further scanning and to prevent any frame shifts. The initiating AUG is found usually as part of a specific sequence called the Kozak sequence, and that sequence is ACC AUGG. So the AUG is part of this longer Kozak sequence, and this is how the ribosome, the tRNA, make sure that this AUG is a start AUG, because it's part of a larger sequence, which would make it a more accurate detection when the scanning of the mRNA takes place. After the GTP bound to EIF2 is hydrolyzed, all initiation factors, including 1, 2, 3, and 4, are released, and they dissociate from the small ribosomal subunit, and the larger ribosomal subunit unites with the small subunit, as I mentioned, catalyzing a GTP bound to EIF5, and that is going to change the structure of the larger ribosomal subunit to maintain it bound to the smaller subunit. Also note that the hydrolysis of the GTP bound to the EIF5 is a proofreading mechanism, so it's an irreversible step because this hydrolysis causes a conformational change in the larger ribosomal subunit that prevents it from detaching from the small subunit. This is the process I just described. This is the small subunit with the mRNA and the AUG sequence, which is part of the larger Kozak sequence, is bound to the initiation methionine. Once this complex is finalized, the 60S subunit bound to EIF6. As we mentioned, EIF6 is bound to the larger subunit to prevent it from binding to a smaller subunit if there is no mRNA present. And then the EIF5 bound to GTP hydrolyzes the GTP bound to it in order to cause a change in conformation in the larger subunit that would make an irreversible attachment between the larger and smaller subunit. Notice that the AUG and the tRNA are at the P site of the ribosome. Also note that the P stands for polypeptide. Once initiation is complete, and that is by the assembly of the ribosome, the elongation phase of the translation of proteins start. As in initiation, elongation requires the presence of elongation factors. At the completion of the initiation, the tRNA methionine bound to methionine is bound to the P site on the assembled ATS ribosome. There's a P site tRNA is linked to the growing polypeptide chain, and that is why it's called the P site, because that is where the polypeptide chain grows. So this is the beginning of the translation elongation part. We have the larger subunit bound to the smaller subunit that is bound to the mRNA, where the AUG sequence of the mRNA is located at the P site of the ribosome, where also the tRNA methionine bound to methionine is located, and where the first amino acid is present at the P site. The second amino acid tRNA is brought into the ribosome as a ternary complex in association with the elongation factor 1 alpha GTP, and it becomes bound to the A site. It's called the A site because this is where amino acid tRNAs enter the ribosome. And then we have a second tRNA bound to another amino acid and bound to elongation factor 1A, bound to the GTP, entering the A site. If the codon matches the anticodon, the process will continue. If the codon on the mRNA does not match the anticodon from the incoming tRNA, then the tRNA diffuses away and another one would come to its location until we have a match between the codon and the anticodon at the A site. So since tRNAs incoming to the A site are GTP bound, they are going to diffuse into the A site, and the next step only happens if there is base pairing between the codon and the anticodon. Once they base pair, the GTP that is bound on elongation factor 1 alpha is hydrolyzed. This causes a conformational change in the ribosome, which causes a stronger bond between the amino acid tRNA in the A site and the release of elongation factor 1 alpha bound to GDP. So this is exactly what happens. Once an incoming tRNA has the correct anticodon, there is a base pair that takes place between the anticodon and the codon. Once that is ensured, the GTP bound to elongation factor 1 alpha is hydrolyzed, releasing a phosphate group and providing enough energy to cause a conformational change in the larger ribosomal subunit that would prevent this tRNA from diffusing out of the ribosome and brings closer the amino acid on the tRNA on the A site with the amino acid on the tRNA on the P site. This ribosomal conformational change, as I mentioned, brings closer the 3' end of the tRNA that is bound to the start codon and the first methionine in the P site. And as I mentioned twice already, GTP hydrolysis is not going to take place if there is no base pairing between the anticodon and the codon at the A site. Once this hydrolysis takes place, the conformational change happens and there's a stronger bond between the tRNA and the mRNA and the ribosome in general, which prevents the tRNA from being diffused away from the ribosome. Note also that GTP hydrolysis by EF1 alpha is another proofreading mechanism that would allow protein synthesis to proceed without having incorrect amino acids bound to the growing polypeptide chain. The alpha amino group of the second amino acid reacts with activated ester-linked methionine on the initiator tRNA, forming a peptide bond. As we mentioned in the previous slide set entitled mRNA decoding, we mentioned that the charging of the tRNA, when we attach an amino acid to its corresponding tRNA, there is GTP hydrolysis, and that is to form a highly activated high energy ester link. The high energy ester link is going to be replaced with a peptide bond between the two amino acids. So this is what happens. The change in conformation of the ribosome is going to bring closer the amino acid on the P site, and since this is the first one, it's a methionine, and the amino acid on the A site. Since this methionine on the P site is bound with a very high energy ester bond with the tRNA, it's going to rather replace this bond with a more stable lower energy bond, forming a peptide bond between itself and the amino acid on the next tRNA, causing the formation of a peptide bond on one hand and the breakage of the ester bond on another. So we have the growing polypeptide chain moving to the tRNA on the A site, and the tRNA on the P site is not charged with an amino acid anymore. This is the translation elongation process. As you see, once tRNA is in the P site, it has the growing polypeptide chain on it, and it has a very high energy ester link binding the growing amino acid chain with the tRNA, and a new peptide bond, which is formed as we described in the first section of this class, is going to form between the amino acid on the incoming tRNA on the A site with the amino acid on the P site that is bound with a high energy ester bond. So this bond is replaced with a peptide bond between the two, and the growing amino acid chain is going to switch to the tRNA on the A site. The peptidyl transferase reaction is catalyzed by the large ribosomal RNA, which precisely orients the interacting atom, permitting the reaction to proceed, which is the transfer of the polypeptide growing polypeptide chain to the incoming polypeptide. After the bond is formed, the ribosome is translocated along the mRNA in a distance that's equal 1 codon, so 3 nucleotides. In order to make sure that translocation is an accurate step, there is the hydrolysis of the GTP in the second elongation factors, which is GTP bound elongation factor 2. Once translocation takes place correctly, the bound GTP is hydrolyzed. This prevents the ribosome from moving along the mRNA in the wrong direction or by a larger or smaller value than 3 nucleotide, which is a codon. This results in the conformational change in the ribosome. This change accompanies proper translocation and results in the GTP hydrolysis, results from the GTP hydrolysis of the GTP bound to the EF2. This is going to cause the tRNAs to shift. So the tRNA I-methionine that was on the P site is going to shift to the E site, which is called that because it's the exit site where the tRNA leaves the ribosome, and the tRNA on the A site moves to the P site. This would happen simultaneously by a second tRNA bound to the polypeptide chain moving to the P site, as I mentioned. This translocation returns the ribosome to the state where the A site is open and allowing new charged tRNAs to come in and check if their codon, if their anticodon matches the codon on the exposed mRNA. And that tRNA is also complex with EF1 alpha GTP, starting another cycle of elongation. So as I mentioned, what happened is that EF2 bound GTP hydrolyzes the GTP bound to it, so we get a GDP plus a phosphate group. This hydrolysis provides enough energy for the ribosome to go back to its initial state where the A site is open and allows more tRNAs to enter and to base pair with the codon using the anticodon on the bottom of the tRNA. Once that is checked, the GTP bound to EF1.GTP is hydrolyzed and the cycle begins again. This is the elongation process in a nutshell. We have incoming tRNAs, base pairing between codon and anticodon, the hydrolysis of the GTP bound to the EF1 that is bound to the tRNA. Once this GTP is hydrolyzed, we get a conformational change that does two functions. One, it prevents the diffusion of the tRNA away from the ribosome. Two, it brings closer together the polypeptides, one on the tRNA on the P site and one on the tRNA on the A site. This closeness causes the formation of a peptide bond instead of the ester bond that links the amino acid to the tRNA on the P site. Once that takes place, the GTP bound to elongation factor 2 is hydrolyzed. This hydrolysis causes the translocation of the ribosome down the mRNA by the value of 1 codon, so 3 nucleotides, and it also causes a conformational change in the larger ribosomal subunit, opening up the A site again for more incoming tRNAs to come in and complete the polypeptide chain. The ribosomal structure has been deciphered using a scanning electron microscopy, and here you can see the 50S subunit, the 30S subunit, and the 70S ribosome that combines the two together. This is the 3D ribosomal structure. This is the mRNA sequence, the 5' end and the 3' end. These are the three ribosomal sites, the A site, the P site, and the E site. And this is the growing polypeptide chain coming out of the P site. Once all the required amino acids are added to the growing polypeptide chain, we reach the last phase of protein translation, which is translation termination. Termination is assisted by factors similar to initiation and elongation factors called release factors. These release factors are employed once the codon on the mRNA is a stop codon. Eukaryotic release factor 1 has a shape that is similar to tRNA, and it binds to the mRNA to stop the codon in the ribosomal A site. Release factor 3 is GTP bound and acts with termination release factor 1 to promote the cleavage of the polypeptide chain from the tRNA on the P site to release the complete protein chain. So this is the ribosome. We have an empty tRNA in the E chain, the P site with the growing polypeptide chain, and an empty A location with a stop codon on it. The release factor 1 bound to release factor 3 bound to GTP associate or base pair with the stop codon because they have complementary sequence to the stop codon. Once it is bound, ERF3 bound to GTP is hydrolyzed to ERF3 bound to GDP plus a phosphate group. This hydrolysis of GTP provides enough energy to release the growing polypeptide chain and to release the larger and smaller ribosomal subunit in addition to the present tRNAs from one another. So the complex is disintegrated and we have our different parts. Most importantly, what we need is the growing polypeptide chain. Similarly to eukaryotes, bacteria have two release factors, RF1 and RF2, as opposed to ERF1 and ERF3. They are analogous, however, the bacterial release factors are analogous to the eukaryotic release factor. The GTPase on the ERF3 monitors the correct recognition of a stop codon by ERF1. Polypeptidyl tRNA bond of the tRNA in the P site is not cleaved until one of the three stop codons is recognized. So otherwise the growing polypeptide chain will remain attached to the tRNA unless a stop codon is released and a release factor is recruited to do so. There are many other release factors. Their function is to basically dissociate the components of the ribosome away one from another. I want you to keep in mind how important the function of GTPases is in the process of protein synthesis. So we mentioned initiation factor 2 GTP, which is hydrolyzed to prevent further scanning of the mRNA once an AUG sequence is reached. We have EF2 bound GTP, which is important to the correct translation of the ribosome along the mRNA. And we have a release factor 3.GTP, which assures correct termination. Note that there are other GTPases, but these are three examples of how GTPases are used to proofread the translation process, because we have already discussed how dangerous and detrimental it is to incorrectly translate a protein. There are many mechanisms used by eukaryotes to enhance the translation process. One of these mechanisms is the simultaneous translation of single mRNA molecules by multiple ribosomes. So we have a single RNA strand with multiple ribosomes bound to it. This is called a polyribosome or a polysome. In addition, there's a poly A binding protein This poly-A binding protein interacts with the mRNA poly-A tail and the 4G subunit that is present on initiation factor 4. This binds to the 5' end of the mRNA. So we have sort of a circle where both the 5' and the 3' ends of the RNA are bound to initiation factor 4. So this is the example. We have the poly-A binding protein on the 3' end of the mRNA. This protein binds the 4G subunit of initiation factor 4, which is also bound to the E subunit of initiation factor 4 that is bound to the cap of the 5' end of the mRNA. This causes a circular stable structure. In addition to that, we can see that there is more than one ribosome bound to the same mRNA molecule, making multiple copies of the same protein from a single mRNA molecule. Another mechanism that our cells use to enhance the translation process is the rapid recycling of the ribosomal subunits after they disengage from the 3' end of an mRNA. If they disengage from that end and more proteins need to be synthesized, these free ribosomal subunits are recruited very quickly and used to translate other mRNA sequences.